Good evening, friends. In today's lecture, we are going to learn about megasporogenesis. What is sporogenesis? As the name itself is suggesting, genesis means synthesis, sporo means synthesis of spores. Of what kind of spores? Megaspores. These are the female spores which are produced inside the flower. Okay. So where the megaspores are produced, megaspores are produced inside the megasporangium. So let's see how does this synthesis of megaspores takes place inside the ovule. So nucleus has this megaspore mother cell which is diploid in nature, so which undergoes meiosis. As you all know that meiosis is a reductional division, so it forms the 4 haploid megaspores. In case of monosporic or polygonum type of embryosac, only one megaspore is functional, whereas the other 3 are non-functional because they get degenerated. So only one megaspore is functional. So after megaspores are haploid in nature. So these megaspores are nourished by nucleus, as we discussed in the previous lecture that nucleus is a nutritive tissue. So it provides it nourishes the megaspores, and or megaspores are also called as a female gametophyte. So this is a megaspore. It undergoes mitosis 1, 2, and 3 divisions, wherein this nucleus, megaspore nucleus gets double. And here you can see there are 4 megaspore tetrads which is formed. Sorry, not tetrads. This is a functional megaspore. It undergoes mitosis 1 and mitosis 2 and mitosis 3 to form 8 celled 8 nucleate cell, 7 cell structure. So you can count in this case there are 8 nucleate and 7 cell structures. So how you can count is see, this the 4 nucleate cells get doubled into 8 nucleate. You can count 1, 2, 3, 4, 5, 6, 7, 8. So this is a 8 nucleate. 7 cell, how you will count the 7 cell is that these 3 are antipodals are 3 number. Okay, and this is a central cell, so it is 4, 5, 6, 7. So it is a 8 nucleate 7 cell structure. Here two polar nuclei fuses to form one central. It gets fused to form the matured embryo sac. Okay, so that's it about the megasporogenesis process. One more thing you should remember is that antipodal's structure of embryo sac is very important to the female gametophyte. It has the antipodal cells which is present specifically in the chalazial end, and they are mainly involved in the they are mainly involved in providing nutrition to the developing embryo. Okay, so this is the filiform apparatus. You can clearly see this. These are the finger-like projections which provides a nutrient supply to the developing egg. Okay, so it is specifically present in the synergids. Spelling, there is some spelling mistake here. synergids. You should leave this a. Okay, it is. I think this is this much is enough. Next you can learn about some concepts regarding megasporogenesis types. Okay, so let us learn about the megasporogenesis type. There are like three main types: monosporic and bisporic and tetrasporic. So monosporic means only one megaspore is functional. Bisporic means two megaspores are functional. Tetrasporic means four megaspores are functional. So uh, monosporic type of embryosac is present in the polygonum, bisporic is present in the allium, and tetrasporic is present in the adoxa. Okay, this is very important. You should remember for the point of examination, or it can be asked in any of the like as an objective type of question in the entrance level examinations. Okay. Megaspores are haploid in nature. Megaspore mother cell is diploid in nature. Let's see how many type of female gametophyte or embryo sacs are present. So firstly, first embryo sac I have already discussed. Okay, this one is a polygonum type of embryo sac, wherein uh, why it is called as polygonum type of embryo sac means it is specifically present in the polygonum. Okay, only one megaspore is functional. That is why it is called as monosporic. And it is a 8 nucleate and 7 cell structure. I have already explained about how you can calculate 8 nucleate and 7 celled in a female embryo sac. Okay, next type is Oenothera. It is also monosporic, but it is 4 nucleate and 4 celled. Allium bisporic, wherein two megaspores are functional. It is having same polygonum type of embryo sac. So okay, bisporic, you can remember monosporic, bisporic and tetrasporic. When compared to the number of nucleus and number of cells, they are very similar. Okay, and only thing is here you should remember the how many megaspores are functional. In case of polygonum or monosporic, only one. In bisporic it is two, and in tetrasporics all the four haploid megaspores are functional. You should remember the examples also. Bisporic, for bisporic, Allium is an example, and for the Adoxa is having the tetrasporic embryo sac. Okay, Plumbago, Penea, Pepperomia, Drusa, Fritillaria. This is very important. It is mostly asked in for it can be asked for a 10 marks question. Okay, for the students who are preparing for bachelor's or masters, or sometimes in UPSC also they can ask this question. So you have to write they'll ask like write a brief note on Fritillaria type of embryo sac. So you should know this is very important. Okay, so it is a tetrasporic embryo sac, wherein it is having the 8 nucleate and 7 cell structure. So I'll explain this in the coming lecture in detail. So just for this lecture, you just remember that it comes under the tetrasporic embryo sac, and it is having uh, 8 nucleate and 7 cell structure. Okay, Plumbaginella is also having the tetrasporic embryo sac. All these are like Polygonum, Oenothera, Allium, Adoxa, Plumbago, Penea, Pepperomia, Drusa, Fritillaria, Plumbaginella. So all these are the plants and are examples for a monosporic, bisporic and tetrasporic embryo sac. So that's it, friends. I hope you have understood this lecture. If you have any doubts, please write in the comment box section. Till that, take care. Bye bye. I forgot to display the MCQs. So these are some of the MCQs which I have explained. These are some of the MCQs you have to solve for the topic which I have explained. So just go through one by one and try to solve it. I hope you have understood the lecture, and you can able to solve all the multiple choice question. If you have any doubt, you please contact me or you can drop me a message. Okay, bye bye. This is very important question. I think you you try to solve it from your side, but I think I feel like it is better to explain this question. So, what is the minimum number of meiotic division required for 120 pollen grains? So, there are 120 number of pollen grains. So, how many meiotic division it requires? So, you know that. Specifically, this is asked with regard to the megasporogenesis process. There's some point which has been missed in the question. Okay, so you just remember that it is related to the megasporogenesis process. So you know that megaspores are four in number, are produced in four in number. So how many pollen grains are produced? 120. How many pollen grains they have mentioned? 120. So divide with that of the 4 megaspores. So 4 threes are 12 and 0 as it is. So there are 30 pollen grains. So there are 30 minimum number of meiotic division which is required for 120 pollen grains. Okay. Remaining questions I think you can solve easily if you have if you have understood the lecture and carefully if you have listened to the lecture. So uh, take care, take care, friends. I'll meet you in the next lecture. Bye bye.